My name is Carl Peters. On behalf of BMG Labtech, I would like to welcome you to today's SLAS tutorial. Today's tutorial will feature the work of Tyler Franklin from OHSU. Tyler has developed a fluorescence polarization assay using a clariostar that has allowed them to study the ubiquitin signaling system. At BMG Labtech, our focus is and always has been on the development of cutting-edge microplate reader technology. One of the more notable developments by BMG Labtech was the first fluorescence polarization capable microplate reader, the PolarStar, back in 1997. Fluorescence polarization has remained an important part of our multifunctional readers, including the clariostar and subsequently the clariostar Plus, both of which have some of the best sensitivity that has ever been seen in a microplate reader for fluorescence polarization. In order to compare microplate reader performance in fluorescence polarization, the accepted approach is to compare variability between replicates. Thus, the limit of detection that is shown here is actually the standard deviation between replicates, in this case containing 1 nanomolar fluorescein. As you can see, no microplate reader is better than the clariostar based on this metric. Without further ado, I would like to introduce Tyler Franklin. Today, Tyler will tell us about ubiquitination and the multiple steps involved in this process. He will also touch on the importance of this modification and how distinct types of ubiquitin linkages have specific impacts on proteins. Further, he will show how better understanding of ubiquitination is needed to improve how we can address diseases associated with alterations in ubiquitin signaling. He will also describe how they use their clariostar to develop an assay that is helping them better understand ubiquitin signaling and all its complexities. Tyler?
Hello everyone, my name is Tyler Franklin. I am a graduate student in Jonathan Prunetta's lab at Oregon Health and Science University in Portland, Oregon, and today I'm going to talk to you about an assay we recently developed called UbiReal, which is a high-throughput assay for monitoring ubiquitination in real time. So what is ubiquitin and what is the ubiquitin system? So ubiquitin is a post-translational modifier that, upon being attached to substrates, can result in multiple distinct cellular signaling outcomes. In order for ubiquitin to be put onto a substrate, it must first pass through the ubiquitin conjugation machinery, which consists of E1s, E2s, and E3s. So the formation of the E1 ubiquitin complex occurs through an ATP-dependent reaction. This E1 ubiquitin complex can then pass an activated ubiquitin onto the E2, and this E2 ubiquitin complex will work with an E3 to ultimately put ubiquitin onto the substrate. The diversification of ubiquitin signaling occurs at this phase, so depending on exactly how ubiquitin is attached onto a substrate will result in different signaling outcomes. So if ubiquitin is attached through lysine 48 residues, it will generate a K48 linked ubiquitin chain, which will be only specifically recognized by ubiquitin binding domains of the proteasomal degradation pathway. So this will target this substrate for proteasomal degradation. Other chain types exist because ubiquitin has 7 different lysine residues as well as a methionine residue that support polyubiquitin formation. And another one of these signals that can occur is ubiquitin chains linked through K63 linkages. And these K63 linkages are again recognized by K63 specific ubiquitin binding domains, which will ultimately result in cellular trafficking of this substrate. In the final phase of this pathway, the deubiquitination machinery will remove ubiquitins from the substrate, which will return a monoubiquitin species, which can then pass back through the ubiquitin pathway. Because of its extensive involvement within the cell, it is perhaps unsurprising that dysregulation of the ubiquitin system can result in numerous disease states. I have a couple of examples, the first being neurodegenerative diseases such as Alzheimer's, which can occur when plaques build up in the cell, while plaques would normally be targeted for proteasomal degradation, a malfunction of the ubiquitin system prevents this degradation, and these plaques are allowed to build up and cause disease states like Alzheimer's. In the case of cancers and autoimmune disorders, you frequently get mutations in some of your ubiquitin conjugation or deconjugation machinery. In the case of cancer, you would get a mutation that results in the ubiquitination of an anti-tumor protein such as p53, thus resulting in more cancer disease states. And in the case of autoimmune conditions, it's frequently observed that a mutation of one of these proteins will result in constitutive activation of the NF-kappa B pathway, which will result in the body sort of attacking itself and an autoimmune state. Because of the extensive diseases associated with ubiquitin, there is a large therapeutic potential for the ubiquitin system. And in fact, there are already a number of drugs that target ubiquitination. For example, perhaps the most famous drug is bortezomib, which will prevent proteasomal degradation from occurring. So bortezomib essentially targets the ubiquitin pathway over here, where it prevents this K48 signal from being read out and ultimately prevents proteasomal degradation. Another example is PYR41, which actually drugs a different part of the ubiquitin system on the other side over here. So what PYR14 will do will prevent the ubiquitin E1 complex from being formed altogether and any downstream signaling from occurring. And then as a final example, we have the MDM2, which is an E3, and the USP7, which is a deubiquitinating enzyme cascade. Both of these are involved in the pathway of p53 degradation, so they are actually drugs that will target MDM2 or USP7 and ultimately decrease the total amount of p53 degradation. Another example is the activation of ubiquitination, which another hot topic is PROTACs. So ubiquitination can be activated using a PROTAC, and a PROTAC, oversimplified, is an engineered E3 ubiquitin ligase that will target a desired substrate, and initiate a ubiquitin signaling event. So then, because of all of these different drugs that can target anywhere along this pathway, what sort of then is the state of the art of monitoring ubiquitination? So currently there are only sort of piecemeal assays that monitor only individual steps along the ubiquitin system. So for example, in ubiquitin deconjugation, there exist a number of substrates that can monitor cleavage by a deubiquitinating enzyme over time, but these assays are not conducive to sort of looking at any other step of the ubiquitin system. And then for the ubiquitin conjugation, there's sort of less standardized assays, and typically what you will see is sort of the gel-based assay, where you sort of get an endpoint is all you really get. So you start here and then you end somewhere over here. So in the gel I'm showing you, we have our purified substrates, E1, E3, E2, and ubiquitin, and then after 5 minutes we sort of get this smear, and that's sort of your readout. So what we wanted to do was sort of exploit this whole system and make a singular assay that could comprehensively monitor each individual step, both for the purpose of simplifying things, but also for the purpose of being able to monitor any potential site where a drug could target along this pathway. So things that we could exploit after thinking about it for a while as well, there's ubiquitin exists in each one of these different steps, so therefore we could probably track the flow of monoubiquitin through the whole system. Another thing that we could work with was that each one of these different ubiquitin complexes actually has a different size overall. So both with the flow of monoubiquitin and the size differences of the complex, we thought, well, we could probably use fluorescent polarization and label this ubiquitin and track it through the system. So how we imagined that this assay would work using fluorescent polarization is that we would be able to use polarized light, and using this polarized light we would be able to excite our labeled free monoubiquitin, and because of its small size and low molecular weight, it would result in faster tumbling in solution, and as a result this would give us a lower FP signal. However, when the labeled ubiquitin was in complex with one of the ubiquitin regulators like an E3, it would be a much larger complex, and as such would have a much slower tumbling in solution, and this would result in a much higher FP signal. And the idea was that with size that this fluorescent polarization signal would be able to scale. And so we had a Clario star from BMG Labtech in the lab, and we began to play around with this idea. So when we tried to use fluorescent polarization to comprehensively monitor the entire ubiquitin pathway, this is the readout that we got, which was pretty exciting. So when we just have our free ubiquitin solution, which is just about 8.5 kilodaltons, we get a relatively lower FP signal around 190. And as soon as we spike in our E1, which is a very large protein, and we form the E1 ubiquitin complex, we get a much larger FP signal of about 245. And then upon spiking in some E2, which would be a lower size, a smaller molecular weight protein, we get a proportional decrease down to about 220. Next, we can spike in our E3, and we get an appreciable increase in FP signal over here, and this will obviously change depending on the size of your E3 that you use. But then one cool thing that we found out is by simply spiking in some unlabeled ubiquitin and generating much larger ubiquitin chain species on the E3, we can get an increase in signal over time as longer chains are built, and you get a slower tumbling complex over here. And in the final step of this, when we added a deubiquitinating enzyme, lo and behold, we could see that this signal would begin to decrease as these signals, large ubiquitin complex signals, slowly were cleaved to free the fluorescently labeled ubiquitin by itself. So we had built sort of a comprehensive assay, but then we sort of wanted to see along each one of these pathways here between ubiquitin and the E1 complex, between the E2 ubiquitin complex and the E3 ubiquitin complex, if we could sort of monitor what was exactly going on between here. And for example, maybe be able to see if a drug could target one of these spots, and if the assay could then be used to characterize that drug. So this first step that we began to look at was actually the formation of this E1 ubiquitin complex. And in order to characterize this, we used a drug that I had mentioned earlier, PYR41, which will actually inhibit the loading of ubiquitin onto the E1. And so using UBReal, pretty reassuringly, we were already able to see that at 33 micromolar of this PYR41, we were able to completely inhibit the formation of this complex, and we could see this. And at our 0 micromolar PYR41, there was complete complex formation, and we were able to get this nice titration in between 0 and 33 PYR41 using UBReal. And we were able to turn this into a nice IC50 plot right here, and able to calculate an IC50 value of 9.15 micromolar. Now this was actually pretty cool because it showed we could use UBReal to characterize one of these drugs, but it was also cool because PYR41 had previously reported low resolution IC50 value of less than 10 micromolar, and this was done using a radioactive based gel. So already with our maybe more elegant assay, we were able to calculate and characterize this drug a little bit better than as previously done. So then it was pretty cool because we had already showed that we could actually use UBReal to characterize this E1 ubiquitin complex. And then next along this pathway, we set our sights on this E2 ubiquitin complex. So it is the case that the E2 ubiquitin, in some cases, the C2 ubiquitin complex can usurp using any E3 at all, and it will attach the ubiquitin onto its target substrate. Now, previously used gel-based assays will have actually used amino acids as a proxy for actual substrates. So that is, so they would put in some free lysines or some free cysteines, and then see if the E2 ubiquitin complex could load that ubiquitin onto those lysines as proxy for a substrate. It is also the case, however, that these E2 ubiquitin complexes will often work with an E3, which will sort of act as an activator and help the E2 ubiquitin complex discharge that ubiquitin onto its substrate. And again, this has previously been done using other assays using amino acids as a proxy for an actual substrate. So then the idea to be able to replicate this assay and make it a fluorescent polarization-based assay using UBReal instead of a gel-based assay was, in this E2 ubiquitin complex, we would have a relatively high average FP, and then upon being loaded, the ubiquitin being loaded onto one of these free amino acids, you would generate a much lower signal, pretty close to the baseline that we see at the beginning of this curve over here. So when we actually tested this E2 ubiquitin complex and the UBReal's ability to visualize a ubiquitin being loaded onto a free amino acid, we used the E2 UBE2D3. And so on this graph, what I'm showing you here is in this light pink right here at 2, we put in some free lysine amino acid in this light pink curve, and upon addition here, you see this slow decrease in signal over time, which shows the loading of the ubiquitin onto the free amino acid as that signal drops. And then over here in this red color, what I'm showing you here is the use of an E3, which again is acting as that activator and helping UBE2D3 load the ubiquitin onto its substrate. And so using E4BU in this red curve over here, adding it in 1 and then adding the amino acid in 2, we can see a quicker discharge of this ubiquitin onto the lysine. And over here in gray, the curve here I'm showing is the use of cysteine instead of lysine, and this transfer happens a bit quicker because it represents sort of a high energy thioester bond transfer. So at this point, we had been able to use UBReal to characterize the E1 ubiquitin complex, and then we had just been able to use UBReal to characterize this E2 ubiquitin complex and its discharge of the ubiquitin signal onto amino acids as a proxy for a substrate. And as I had mentioned, this was previously done using a more tedious gel-based readout, but using our fluorescent polarization assay, we were able to monitor this same discharge with a Z prime value of 0.83, as well as doing so in a 384 well high throughput format. So having done these two steps, we then moved our sights on to the next step, which was that E3 ubiquitin complex. And as I had mentioned earlier, we were able to use a little trick, which was spiking in some unlabeled wild type ubiquitin into this reaction, which could then support the formation of not just the monoubiquitinated E3, but the generation of the generation of polyubiquitin chains onto the E3 over time, giving us a larger increase in signal. So what we wanted to ask next was if we could tease apart what was going on with this larger E3 ubiquitin polyubiquitin complex and see if we could visualize what sort of chain linkages these were. So in order to do this, we employed a set of ubiquitin mutants. And so these ubiquitin mutants, while ubiquitin normally has seven lysine residues that can support chain formation, if you go through and mutate all of them away except for one, you generate your what are called K-only mutants. And so each of these only has one of the lysine that can be used to support chain formation, or our 0K lyseless mutant, which has no lysines and cannot support any sort of ubiquitination at all. And our idea was we could go ahead and spike these in and see if using these we could either enhance or degrade how much signal is made here. And just as a brief reminder, we are interested in what sort of chains are made because they are critically important to what cellular outcome happens in the end. So more in depth here, the idea is if we used a K63 specific E3 ligase and we fed it one of these K-only mutants that does not have a K63 mutant, then we would only be able to support a monoubiquitinated E3. But if we fed this same K63 specific E3 ligase with the mutant that does have a K63 lysine or wild type ubiquitin itself, we could then support an E3 ubiquitin that could write ubiquitin chains, both free ubiquitin chains, and put larger ubiquitin chains onto itself, again because it's a K63 specific ligase that needs this lysine to make larger chains. And so the idea was that this ultimately would result in a much higher average FP signal if we have a K63 specific ligase and use a K63 mutant, whereas if we fed the K63 specific ligase one of these other mutants which couldn't support K63 ligation, you would get a smaller overall species and you would get a lower average FP signal. So when we did this, we actually did use a K63 specific ligase, which is known as NED4L. And when we used NED4L and we gave it a wild type ubiquitin, we would generate 100% of our signal here. And if we use that K0 or lyseless mutant, which can only support monoubiquitination of the E3, we then get a signal of around 50%. And then the same thing with this methylated ubiquitin, which also has all of its lysines as well as its methionine, chemically altered so it can't support polyubiquitin formation, we get about the same signal. And down here, I'm just showing you the control of the fluorescently labeled ubiquitin by itself. So again, this higher signal here is likely describing the formation of these free ubiquitin chains, as well as the E3 conjugated with much higher order species of polyubiquitin. Whereas down here, we are only able to get that monoubiquitin formation, and because of our mutant, we can't support polyubiquitin formation. And so then now, again, NED4L being a K63 specific ligase, when we use our K-only mutant panel, we get this graph over here, which was pretty exciting, because what it showed was that as we had thought, that K63 only ubiquitin would be the only one that could support the same sort of ubiquitination events as the wild type ubiquitin. And so this K63 only mutant, which is the only one that has that K63, was able to generate roughly the same signal as the wild type mutant, whereas all of the other K-only mutants, which K48, K33, K29, K27, K11, and K6, were only able to generate about a 40%, somewhere between a 30 to 50% signal over here, meaning that it was likely telling of this monoubiquitinated E3 giving a much lower FP signal. So this was pretty exciting, because it meant that we could use UBReal to visualize exactly what chain linkages were being formed. So up till now, we had been able to use UBReal to look at each of these different ubiquitin conjugation events. Most recently, we were able to look at the E3 ubiquitin complex and chain formation over time. And using our mutants, we were able to see the chain-specific formation of ubiquitin linkages, which was very exciting. And we had one final question for UBReal, and that was if we could use UBReal to characterize deubiquitination events. So in order to do this, we used a principle known as ubiquitin chain restriction, or UbiCrest, which is based on the principle that, like E3 ubiquitin ligases, which will only generate specific signals, dubs also have the same sort of specificity and will only cleave certain signals. So using our pool of NED4L generated ubiquitin species, we could then toss in different kinds of deubiquitinating enzymes to see what sort of readout we could get. So if we used an unspecific deubiquitinating enzyme, you would expect that all of the ubiquitin signals would be cleaved, and then we could generate a signal back to the baseline that we see at the beginning of the assay. If we use what's known as a K63 exospecific dub, which is a deubiquitinating enzyme which only cleaves K63 ubiquitin signals, but is unable to cleave the terminal base ubiquitin, which resembles a monoubiquitinated species over here, then you would expect to see sort of a middle of the road FP. And if, on the other hand, we used a K48 specific deubiquitinating enzyme, you would expect to see about no change in the FP, only in the case of using NED4L generated ubiquitin species. And again, this is because the NED4L is only going to generate K63 specific species, and meaning it can only be cleaved by a K63 or an unspecific dub, and a K48 dub should not be able to cleave anything on anything that is generated by NED4L. So when we did this using UBReal, again using our pool of NED4L generated ubiquitin species and treating it with these different dubs, we obtained this graph over here on the right. And the first deubiquitinating enzyme we used was USP21, which is one of those non-specific dubs for both mono and polyubiquitin species, and we could see that 100% of this signal would be cleaved down to about 0% over time, as you would expect for one of those non-specific dubs. Then we used a couple of other different dubs, the first being AMPH, which is specific for that polyubiquitin K63 ubiquitin species. And what this showed us was that this signal, in this light pink here of AMPH, that it would cleave the signal to about 40%. And this was pretty cool, because what it told us that about 60% of this signal that was generated by NED4L was coming from the generation of polyubiquitin K63 species, and that the remainder was likely this monoubiquitin terminal species that, again, AMPH is not able to cleave. And then using a couple of other dubs as controls, OTB1, which is specific for 48 linkages, and then Otrlin, which is specific for methionine 1 or linear chains, neither of which should be in our NED4L generated pool of substrates. We could see that the signal would not be cleaved, and we would get about 100% signal at the end of the assay still. So in the end, we were able to obtain a first-of-its-kind comprehensive assay that was able to monitor each individual step along this ubiquitin system pathway. So we're able to look at the formation of the E1 ubiquitin complex and the drugging by PYR41. We were able to look at the amino acid discharge by the E2 ubiquitin complex. And excitingly, we were able to look at the chain-specific formation of polyubiquitin signals by E3 ubiquitin ligases, as well as the breakdown of those polyubiquitin signals by deubiquitinating enzymes, all using the same assay. So you can imagine how this could be particularly powerful because it's a single assay that you could then test any sort of kind of drug that would target one of these individual phases along the ubiquitination pathway. You could also imagine using activators, which could then increase the amount of ubiquitination or the amount of deubiquitination over time, again using the same assay. Another powerful aspect of this assay is that it lends itself, because of its fluorescent polarization protocol, to a high throughput format. And so in the end, we were able to publish this assay in Frontiers in Chemistry last year, and then we were able to also write an application note for BMG Labtech. So I'd like to time to thank BMG Labtech for the opportunity to present, as well as to write that application note. I'd like to thank the other members of the Jonathan Pernatal Lab, including Justine and Cam. I'd like to thank my funding from the NIH, as well as our lab's funding from OHSU. And thanks everybody for your attention.
Thank you, Tyler, for that excellent talk. We're happy that the Clariostar was able to help you to develop this very useful assay. Before we close, I would like to take a moment to tell you a little bit about one of our other excellent fluorescence polarization readers. That being the Ferrostar FSX, which is the latest edition of our high-throughput screening instrument, the Ferrostar. You will probably recognize this comparison of different readers based on LOD, which is a report on the standard deviation or variability that is seen for a constant sample of fluorescein. Again, the Ferrostar FSX is better than the readers by the other readers in this group. You'll probably also notice that the Ferrostar FSX has the same LOD as the Clariostar. So really, at this point, BMG is in competition with ourselves for the best fluorescence polarization readers. So why would you choose the Ferrostar FSX or the Clariostar? Well, it really depends on your needs. The Ferrostar FSX is designed to be our high throughput screening instrument and therefore has the best sensitivity and the best speed. It can do this due to a number of different factors, but one of the major players is these assay-specific modules that are shown here, as well as the fact that it has matched dedicated PMTs. But the primary difference between the Clariostar and the Ferrostar line is the fact that the Ferrostar has simultaneous dual emission. So what this means for fluorescence polarization is that both the parallel and perpendicular emission signals can be read simultaneously. Therefore, the read time that you will have with the Ferrostar will be half what you see for the Clariostar. I would just like to thank you all again for your attention, and of course, thank you to Tyler for an excellent presentation. And if you are interested in BMG Labtech's lineup of readers, including the Clariostar and Ferrostar, please come and see us.